This is a list of all the science and tech concepts that were asked in the UPSC Prelims 2023 paper, either as a standalone question or as part of an option to a question. We'll go through these topics one by one and develop a comprehensive understanding for each of them. Welcome to UPSC Anna. This is our Learn Science and Tech through PYQ series, where we're going to cover every science and tech topic that has come in the previous year's prelims papers, beginning with Prelims 2023. If you find the video useful, consider liking and subscribing and all that stuff, as it really helps us out. Let's begin. Extremophiles are very interesting organisms. These are the organisms that can live in very extreme conditions such as very high heat, very cold, or in very acidic or alkaline environments. Some bacteria live quite comfortably in extremely high temperature environments like in hot springs and hydrothermal vents on the ocean floor. We call them thermophiles because they thrive at high temperatures. Similarly, there are microbes in Antarctic ice that can survive in very low temperatures. These are called psychrophiles or cryophiles. Extremophiles that live in highly acidic environments are called acidophiles. These organisms thrive in acidic conditions, where the pH is very low, usually below 3. The study of extremophiles, called extremophilology, not only tells us how adaptable life is, but also suggests that life could exist on other planets, where conditions are extreme. It also helps us understand how life on Earth might have originated and how early life forms evolved in these harsh conditions. This information is also quite useful in space exploration and astrobiology, as it gives us hints about what kind of environments we should look for, the possibility of life on other planets. The Wolbachia method is a very interesting and innovative approach used for population control of certain types of insects, especially mosquitoes. This method is particularly useful for preventing the spread of diseases transmitted by mosquitoes, such as dengue, Zika, chikungunya, and yellow fever. Wolbachia is a type of naturally occurring bacteria found in many insects, but not naturally in many mosquitoes, such as Aedes aegypti, which is known to spread diseases like dengue and Zika. In this method, scientists introduce Wolbachia bacteria into Aedes mosquitoes under laboratory conditions. When these Wolbachia-infected mosquitoes are released into the wild, they breed with the natural mosquito population. Since Wolbachia induces certain types of reproductive effects, such as cytoplasmic incompatibility, the offspring produced by infected females also carry this bacteria, while the eggs of non-infected females often aren't viable. Thus, the population of Wolbachia-infected mosquitoes naturally increases over time. A major benefit of this is that Wolbachia-infected mosquitoes do not transmit viruses that spread diseases like dengue and Zika as effectively. This method not only controls the mosquito population, but also reduces the spread of these diseases. The special feature of the Wolbachia method is that it is an environmentally friendly approach, avoiding the use of chemical insecticides and preventing negative impacts on the ecosystem. Let's start from the basics and build up our understanding of aerial metagenomics. A gene is the basic unit of heredity, which is essentially located in our DNA. These genes help control all the features and functions of our body. Imagine, just as recipes are to a recipe book, genes are to our body. Now, if every gene is a recipe, then the entire recipe book can be referred to as the genome. This is the set of our entire DNA, which we inherit from our parents. The genome essentially includes all the genetic information of an organism. Genomics is the field that studies genomes. This means scientists in this field try to understand how our DNA defines us and how different genes influence our health, appearance, and behavior. Metagenomics is a bit more interesting. It's the study of DNA found in a particular environmental sample. This means if we got a handful of soil, then the study involving all the DNA from the microorganisms present in it would be called metagenomics. It tells us about the diverse life forms present in a particular environment. Now let's talk about aerial metagenomics. In aerial metagenomics, scientists collect DNA from air samples and then analyze them. This helps us understand which microorganisms are in the air. This gives us information about the quality of the environment, pollution, and even patterns of disease spread. Before diving into microsatellite DNA, let's discuss some basic peripheral concepts. First, let's talk about nucleotides. These are the building blocks of DNA. The structure of DNA comprises 4 types of nucleotides: adenine, thymine, cytosine, and guanine. These nucleotides arrange in a specific sequence to form our genetic code. Speaking of genes, a gene is a segment of DNA containing specific instructions that code for a particular trait, such as the color of your eyes or your height. These genes are made up of specific sequences of nucleotides. DNA is the molecule that carries all our genetic information. It has a double helix structure where pairs of nucleotides are arranged like steps on a ladder. Every gene is a part of our DNA, and all the functions and characteristics of our body are determined by these genes. Microsatellite DNA, also known as simple sequence repeats or short tandem repeats, is a type of DNA sequence that repeats within our genome. These are short sequences that repeat one after another multiple times. These repeats are very common in our DNA and vary slightly in length and sequence among individuals. These repeats create unique patterns in our DNA which have several applications in identification, research, and conservation. Let's discuss some applications. Forensic experts use microsatellite DNA to identify suspects from biological samples found at crime scenes, such as hair or blood. Since every individual's microsatellite patterns are unique, this method is very accurate. Due to the uniqueness of microsatellite analysis, it is also used in paternity tests. Microsatellite DNA plays a significant role in studying the populations of wild animals and plants. It helps scientists understand species diversity, population structure, and breeding patterns. Microsatellite markers are used to understand and improve the genetic diversity of plant breeds. This leads to improvements in crop yield and disease resistance. Microsatellites are used as genetic markers for certain diseases. Researchers can use these patterns to identify predispositions to some diseases, and they are also helpful in personalized medicine. Bioplastics and biodegradable plastics are related concepts but are not the same thing. Each has distinct characteristics based on their origins, production processes, and environmental impacts. Let's understand in detail. Bioplastics are derived from renewable biological sources, such as vegetable fats and oils, cornstarch, straw, wood chips, food waste, etc. Not all bioplastics are biodegradable. Bioplastics include both biodegradable and non-biodegradable varieties. For example, PLA, polylactic acid, is a biodegradable plastic made from cornstarch, while biopolyethylene, made from sugarcane, is not biodegradable, but is derived from a renewable source. The purpose of bioplastics is reducing the reliance on fossil fuel resources for plastic production and reducing carbon footprint. Biodegradable plastics can be made from both renewable sources, that is, bioplastics, and non-renewable sources, that is, petroleum-based sources. These are specifically designed to break down into water, carbon dioxide, and biomass with the help of microorganisms under certain conditions. The time and environment required for degradation can vary significantly. Biodegradable plastics includes plastics that are both bio-based, like certain types of PLA, and those that are not, such as oxo-degradable plastics, which are petroleum-based plastics with additives that promote oxidation and fragmentation. There are some key points of differences between bioplastics and biodegradable plastics. First is that bioplastics are defined by their renewable biological origin, while biodegradable plastics are characterized by their ability to be broken down by microorganisms. And secondly, not all bioplastics are environmentally friendly in terms of degradation, and not all biodegradable plastics are made from renewable sources. The benefits and drawbacks of each type depend on various factors including production processes, disposal methods, and specific applications. In essence, while there is some overlap, that is, bioplastics can be biodegradable and some biodegradable plastics are bioplastics. The two terms refer to different attributes. Bioplastics are about where the material comes from, and biodegradable plastics are about where the material ends up. Now try solving this question and post your answer down in the comments. You can find these slides and all our other UPSC related content on our Telegram channel. Links in description. Like, subscribe, and all that stuff if you found the video helpful. And make sure to supplement any videos you consume with active self-study. Remember, learning is doing, not just viewing.